just to try and emphasize how powerful this is, the number of experiments we can do within a year is roughly 3 centuries of future wet lab experiments. So it's like getting into your DeLorean, going to 2325, getting out the DeLorean, like reading the results, right, like the best results, coming back to now, right, and doing that in the wet lab. And that's sort of the power of like having things in the computer.
Welcome back to the Health Longevity Secrets Show, where we push the limits of human potential and unlock the secrets to our health and longevity with your host, Dr. Robert Lovekin.
Today's guest is Dr. Daniel Ives, founder of Shift Bioscience, and his story is a wild pivot from the classic pick a hallmark of aging science to letting the data drive everything. We talk mitochondrial DNA, why his original hypothesis didn't survive the epigenetic clocks, and how Shift now uses single-cell aging clocks plus virtual cell models to run literally centuries of experiments in silico. And then it gets even crazier. Single genes that can rejuvenate cells without pluripotency, plus real translational targets like fibrosis and age-related hearing loss. Buckle up. This is the leading edge of the longevity revolution.
And now, please enjoy this week's episode.
Hey, Daniel, welcome to the program.
Great to be on, Rob. Great to be on. I appreciate the invitation.
I'm so excited to talk with you today. You know, you have an amazing journey from studying mitochondrial DNA mutations at Cambridge to founding really one of the leading companies in the longevity space, Shift Bioscience. And before we dive into that, let's maybe take a moment and tell us a little bit about your journey, how you came from where you started to here.
Yeah, so this is I think a common story. I watched a TED talk by this scientist called Aubrey de Grey, and just captured my imagination. You know, just such a spectacular talk around the time I was graduating. And he said two things. Firstly, let's do something about aging, right? Let's not just assume it's off limits, right, to intervention. Like, don't put it on this pedestal. Let's get off our seat, start doing something. And then immediately afterwards, which is key, he said, here's a framework for aging, like breaking it down into digestible pieces, which sort of became the hallmarks of aging, right? This framework. And he just lists them out and he says, here's an order of hallmarks. And I bought his book immediately, read the book. I think in the book it said the mitochondrial genome is one of the hardest things to solve. So I was like, hell yes, that's what I'm going to try and do. I'm going to try and solve this genome. And he'd sponsored a PhD in University of Cambridge in the UK. And so I sort of reached out to the PI that was collaborating with Aubrey. And unfortunately, that project had been shut down. I didn't have a plan B, and then I found a plan B within the lab and got to work trying to solve aging. And I believed what I was working on was the sole cause of aging, and it would fix it in its entirety. So when I did solve this tiny little problem, I had that feeling like I'd solved aging. And after that, I was determined to take that to the world. And eventually that became Shift Bioscience. And the first thing we did as a company was disprove the hypothesis behind the company. So we showed this little thing was just a little thing. I mean, maybe it will be a bigger thing in the future, but it was one part of the picture, and ended up pivoting the company hard into machine learning as the route to the biology that we want to manipulate to have the biggest effect size on things like lifespan eventually. Um, yeah, and along the way, just spending my own money, spending other people's money. Um, eventually bring an investor on board, staying very little, then getting very big very quickly, and just you know, all of the faculties you have to develop to sort of manage a team at a higher level, having to communicate, right, having to learn how to do that. Um, but yeah, it's a pretty crazy journey trying to confront this thing. It's like a perpetual learning experience, right? The more you see of it, the more you realize is ahead, and you don't want to let that put you off, right? You want to reel in the finishing line every opportunity you can, but then you've got to try and kill what you're doing all of the time, not to waste time. Um, so I just hope I survive the process.
That's a great introduction. It's funny. I didn't realize that Aubrey's talk influenced you. It's what a coincidence because I was up in Monterey and I was going to this new conference, or it wasn't new. It was new to me. It was called TED, and
Yeah,
they had these speakers and. I remember sitting in the audience when Aubrey gave that talk, and it was a great talk. It was mind blowing, and it's so funny you listen to it as well. I mean, that's a great conference. I think of the same conference or one of the two of the years before or after they had another guy from he was a guy who'd started PayPal and he had an electric car that he brought there that he was showing to everybody and named Elon Musk and he was trying to launch this Tesla thing and but that's fascinating with Aubrey. He's such an inspiration, and it's amazing to think that yeah that he's influenced the course of your career that way, and now you're taking up the torch and going on. Of course, Aubrey's still doing great things too, but
yeah.
it's nice to see that. Well, I'm really looking forward to getting into a little bit of details on this, and I don't want to lose some of our audience who may not know some of the technical stuff. Let's just start with at the beginning, kind of define some of the terms, what we agree on, like aging and longevity. So, like, what what is your how do you think about longevity and aging? Is it just, oh, our cells wear out and it's just inexorable time, or is it, hyperfunction like Blaglasconi and others, or is it something else? Why do we age? What causes aging? What are we trying to fix here?
Yeah, so I'm sort of shaped by my personal experience, which is I sort of dived hard into hypothesis-based science. Aubrey, hallmarks of aging. I chose the hallmark I think was most causative, right, based on the evidence. I worked on that very diligently, and then I sort of, I tried to go as far as I can, and ended up disproving this was a causal factor, right, at least with the technology available. So I ended up basically hanging up my boots as a hypothesis-driven scientist and embracing the world of unbiased science where you use very rich signatures like single-cell transcriptomics or any types of omics, and you let the data take you to the answer because the cell is just this unbelievable physical computer, right? Like, and if you've done biochemistry, biology, it's like a Lord of the Rings epic. Like you read the first chapter, it's just like immunology, and then there's like basic biochemistry, and it's like you're learning all these characters and how they behave, and that's just what we know about the characters and how they interact, and there's everything that remains. So the idea that you could just understand a part of that infinite complexity, is sort of, I think what tends to happen is you create a comfort zone of mechanism that's still surrounded by infinite black box. And I sort of found out the hard way because I was, risking my own money and time. So I fully embraced unbiased science, and that's, now it's machine learning. So I'm going to give you a really terrible answer, which is I don't actually worry about exactly how it's happening. We just have certain metrics for being older, being younger, which are unbiased metrics, and we sort of we they're the Lord, right? The metrics are the Lord, and they might lead us down a dead end, right? And we fully acknowledge that, and that's a risk. But ultimately, they're like a compass that we're so lucky to have. And so for me, I think what will happen is we'll find this biology that does this remarkable stuff, and it'll be like finding a UFO in your backyard, and then we'll have to reverse engineer it, and that will reveal the elegance and the majesty of the rejuvenation process. But that's the way around I see it. Maybe when we get close to the answer, we'll start mechanism will start catching up, but I'm not putting mechanism in the way of utility, right? So I just want to get to utility, let the machine learning take us there, and then we can enjoy the story at the end. I don't know if that's the correct approach or not.
I love that. I love that sort of. I think we could all benefit from taking that strategy to other things we do in our lives and other approaches to thing. In other words, look at almost first principles and look at the what the evidence produces and then have the hypothesis afterwards. And really, it sort of turns the science a little bit on its head. Back in the day, we have to be hypothesis driven because, doing one experiment was a big deal. It took a lot of time and money. But now with AI, we can literally generate, many hypotheses in parallel and basically let mechanisms drop out of that.
Yeah,
Right?
just to add sort of add one line on top of that, which is with these systems, we can actually do every experiment we want to do. Right? This is the great thing. We don't have to be choosy, right? Like we've only got a certain amount of time, so we can only choose one experiment and then we go. We've got such experimental power now that we can effectively do every experiment and then just choose the best results to take into the wet lab. And then we basically just confirm on the back end. So um, this sort of stuff wasn't possible until very recently. It's really a product of the sort of the compute and the models that sit inside that compute. So I'm very lucky that this has happened around the time I was trying to solve this problem, right? Without these capabilities, we'd be struggling away for like many moons, but we're able to find stuff much sooner.
Yeah, and we're going to dive into that at when we talk about specifically what you're doing at Shift Bio Science. But before we leave this, maybe what was the failed hypothesis that you tested? It was around mitochondria, right? Your initial
Yeah.
thing in grad school, I guess.
Yeah, so sort of basic biology of the eukaryotic cell. We are sort of multicellular version of the eukaryotic cell, but each cell has two genomes. So there's the famous genome, which is the nucleus, and there's the less recognized genome, which is the mitochondrial DNA. And over time, most of the genes from the mitochondrial DNA have sort of migrated west, right, into the nucleus. They found a safer home in the nucleus, but these 13 genes that make 13 proteins persist in the mitochondria, and they're encoded on the mitochondrial DNA. And it's actually a whole genetic system separate to the nuclear genetic system. It's like running it's like running a Mac inside a Windows operating system, right? It's like sort of bizarre, and it's very expensive to do this as well. Like you need a whole set of new ribosomes and tRNAs to run this system. Um, but that genome's very important, and the hypothesis I was working with was mutations in this genome are the underlying driver of aging, and there's some very provocative evidence that this was the case. So, for instance, if you delete the proofreading domain in the polymerase that reproduces this circular genome, you basically create about 2,500 times the level of damage every time you replicate the genome. So it's like just firing damage into this tiny little genome, and you create a very comprehensive human aging phenotype in a mouse. So this mouse loses hair behind its neck, gets kyphosis of the spine, it gets enlarged heart. This is actually more like a human aging phenotype rather than a mouse aging phenotype. And I think there was like a Nature front page at the time, like mutator mouse aging solved. Like paraphrase, right? They were like, we solved aging. And I'm reading that and I'm like, hell yeah, I'd already chosen that and it's confirmation bias. So, we got hold of one of these mice and I had this type of therapeutic and we put it in the mouse and then we managed to slow aging and partly reverse aspects of aging in this mouse, only confirming my belief I'd solved aging. But this all came to a screeching halt with the discovery of the epigenetic aging clock. So Steve Horvath, discovered this very robust multi-tissue biomarker of aging within the methylome. He benefited from these huge databases full of omic data, like methylome data. But these clocks were like the major breakthrough for the field, like having a measure of aging that you can experiment against instead of having to run huge lifespan experiments. It was like a really big step forwards. So I wanted to use this clock to prove to the world that damage to the mitochondrial genome was the major cause of aging, and by affecting that damage, I would, stop, slow, reverse aging. And what I showed was that by changing this damage, it had zero effect on the clock. So the clock was like blasé, right? Like we put the damage up, it didn't spin faster. We put the damage down, it didn't spin slower. So, you know, this damage was creating an aging phenotype, but that aging phenotype wasn't driven by an acceleration of physiological aging. It was driven by a genetic change, specifically to the mitochondrial genome, that has the common phenotype with physiological aging. So I had a lot of faith in this biomarker. Like I was hell bent that this biomarker was the compass we need to follow. So for me, if we didn't get the answer, that was a reason to move away from this flawed approach and do something else. And that's exactly what happened. So the results came in, and we basically fully embraced aging clocks as the route forwards. And we basically, we sort of doubled down on clocks, make them high throughput, do CRISPR screens where we can scan all biology, looking at the clock, seeing where it's responding. And there's, basically a supercharged version of that, which we can do in the computer now, run all experiments, see what the clock's doing, bring the winners into the wet lab, and that's how we find the genes.
Well, and let's put a pin in the clocks too, because I want to come back and talk about clocks when we talk
Yeah.
about Shift Bio and specifically what you're doing, and that's so important. That's fascinating because, like, there's some people believe that with mitochondrial DNA mutations and impeded energy metabolism, right, the cell just can't do that. The impeded energy metabolism drives epigenetic changes on the epigenome in the nuclear DNA, forcing it to select more energy efficient functions. I mean, it just makes sense. If you can't operate, if you're not making energy, you'll select, you won't do certain things. But you're saying that didn't really pan out much, that there weren't epigenetic signatures that corresponded to the mitochondrial DNA changes, really, right?
Yeah, so within the narrow window of the mitochondrial DNA, and basically this, I think it's, 2,500 times more damage, we didn't see the clock respond. But then within a broader window, which is like mitochondrial numbers or mitochondrial function, there is a relationship. So Ken Raj, who's now at Altos Lab, he published a study which was assessing all of the different hallmarks of aging and how they affected the clock, right? He wanted to sort of systematically change those hallmarks in the positive and negative direction and see which hallmarks were driving this clock, maybe like basically which hallmarks are more important. And he did find a relationship with mitochondria, and it was a reciprocal relationship. So I think he used beta fibrates to boost the biogenesis of the mitochondria, and the clock ticking slowed down. So it sort of slowed the rate of aging or epigenetic aging. He then used another reagent which is called CCCP, which basically it depolarizes the inner mitochondrial membrane, and this basically interrupts function, and it made the clock spin faster. So you have this reciprocal relationship, right? Like make more mitochondria, there's more mitochondrial function, clock ticks slower. Disrupt mitochondrial function, the clock spins faster. So within the narrow window that I was asking my question, which was just mutations, there wasn't a relationship. If you just go a little bit broader, there was a relationship. So it just shows you that sometimes you miss the bigger picture and you make a conclusion that's a little bit too, it's ultimately a false conclusion. There is mitochondrial mutations in isolation didn't have a link to the clock, but then when you consider mitochondrial numbers, that did have a relationship. So maybe there's a threshold effect with mutations where they get bad enough, starts impacting function like CCCP, and then you'd see a relationship. We didn't encounter that in these mouse experiments we ran initially.
Oh, okay, so the pure mitochondrial mutation didn't show that association, but if and that might be due to basically number size, the quantity of the mutated ones or the percentage of the mutated ones and all. Because I'm just thinking of other people in the mitochondrial transplantation space, like our mutual friend Tom at Matrix Bio and Natalie at Minovia and several other people. They've all been on our podcast talking about mitochondrial transplantation as a rejuvenating theory. So there's still maybe you're not discounting that. It's just purely the
Yeah,
mutations itself as an effect, right?
yeah. There's a basis for increasing the number of mitochondria as a means to slow down the ticking of the clock. So that is one of the findings of this paper from Ken Raj. That if you boost the numbers of mitochondria, you can slow down the rate of aging according to these clocks. So
Interesting.
I think
Yeah.
mitochondria is certainly part of the picture. but more at the macro level, not at the micro level of the genome, at least, within the boundaries of the experiment we did.
Yeah, yeah. So, okay, well, good. Not purely the mutation. Okay, so then shift over. One other thing I wanted to talk about was partial epigenetic reprogramming, which is in the news with, sort of the classical approach with Yamanaka factors. Um, what is that exactly? And what are the limitations of that approach compared to what you're doing?
Yeah, so start with your first question. What is it? Like, what is partial reprogramming? So we know precisely zero about it, except there are four factors called Yamanaka factors. They're transcription factors. When you express four of them all together, they can turn adult cells into stem cells, and in the process, they reverse age by almost every measure, including the epigenetic clock. You can substitute, well, you can minus c-Myc, which is one of the four factors that's an oncogene, and you can maintain rejuvenation even with a subset. Um, but the biology that sits underneath these factors in terms of rejuvenation is a mystery, right? It requires biology to be mapped in a way that's quite difficult to do. So yeah, I mean, what's wrong with these factors? Maybe let's just start with that, right? What's wrong with them? It's actually what was right with them. That's the thing. What's wrong with them is what was right with them. So what was the original experiment? Yamanaka was trying to turn adult cells into stem cells because embryonic stem cells were not readily available to researchers. So the idea you could just make them right from a readily available source was like that was a real research area that could do a lot of good. And so he fully optimized his experiment to find genes that make adult cells into stem cells. That's the feature he was optimizing. The bug was rejuvenation. So the rejuvenation was discovered quite a while later, and that is the bug, right? That remains the bug of the Yamanaka factors, not the feature. So you can try and optimize for the bug, which is called partial reprogramming. Yeah, like let's just use it for 2 days at the weekends, right? Let's use it for the weekends, and we can actually extract a positive effect out of these factors. Like that's the therapeutic window. And there's certain use cases where there has been a safety case that's been built around using the factors. I think in the eye, certainly, the eye is one example. I think Yuri Daigan from Youth Bio, there's sort of a case for the brain. It's non-proliferative, so it's not as dangerous to factors. But ultimately, these factors, we're still optimizing the bug when we're talking about rejuvenation. So the interesting question is like, what is the rejuvenation biology? Because if we could learn about that, we could double down on the bug, optimize for the bug, like directly optimize for rejuvenation instead of just optimize for the bug, which is very hard to do. So that's ripe for discovery. Like how do we approach that problem? Like which tools do we need? Yeah, what do we find? Like, do we find anything that's better than Yamanaka factors? Does it need to be combinations? Could it be inhibition? Does it have to be overexpression? Like there's this whole world that needs to be explored. And so, we explore this world. We're finding some very interesting things. Others are exploring this world. They're also doing great work. Um, but the great news is there seems to be biology beyond the Yamanaka factors that's involved in cell rejuvenation.
So even and I think we're all aware of Dave Sinclair's work with the trial going on in Boston, like you say, in the with the certain types of age-related glaucoma and NION in humans, the first clinical trial. And they're using OSK, right, the subset of Yamanaka factors, and hoping to reduce the pluripotency effect of that, and the teratomas and the all that things. But you're saying that really doesn't do it. And we're going to talk about in a minute your approach, which is really interesting from, again, first principles, kind of from the AI and how that drives it, right?
Yeah, so I think David does have a case. He does have a case in the eye. So OSK can be used with a broader therapeutic window than the four factors together. And my understanding is they built a sufficient safety case around this use case in the eye. So I think there's a good chance they do something very positive. My only concern is that they're still using factors that were optimized to be pluripotent inducing factors, right? They were designed to make stem cells. So I think they you can optimize for the bug and get something out of that. Um, but these genes were always designed to do something else. That's my only bugbear, right, with the use of Yamanaka factors. I'm excited because it's the first trial using cell rejuvenation as the mechanism of action. Like it's the first one to sort of break into clinical development, and yeah, that's a great thing, right? It's a long time coming, but I don't think I don't think that's the iPhone 10. I think that's the iPhone 1, and the iPhone 1 was never designed to be an iPhone, right? It was sort of massaged. Maybe it was like a power tool, right? And it was like adapted into a phone. So, I think it's good it's happening, but there's a lot more we can do just by understanding what rejuvenation is. And, iPhone 2, 3, 4, 5, I think there's a lot of headroom to do much, much more spectacular things.
Yeah, we're just at the cusp of this revolution, and it's such an exciting time to be alive. There's no other time in history I think like this, and certainly no other time I'd want to be alive. It's every day is new stuff happening. So let's pivot to AI and talk a little bit about that. And everyone talks about AI, the revolution. It's happening. Gold. Math Olympiad gold medals are now, won by AI, and the benchmarks of these frontier labs keep getting moved and moved and moved. Eventually, in many areas, already surpassing humans, but it's going on. So at Shift Bioscience, you're using machine learning to uncover these true drivers of aging. Can you break that down a little bit about how you approach it? It's fascinating.
Yeah, so we use narrow AI. So this is, by design, looking at sort of a very narrow problem, and we're like pummeling that problem with GPUs, right? It's like you just keep throwing the GPUs into the problem. So the narrow AI, there's two types of narrow AI that we're working on, and one is an aging clock. So the original aging clocks were actually one of the early fruits of machine learning applied to biology. It's basically been impossible to find a biomarker of aging that's high accuracy and works across tissues. But within the methylome, if you take all that methylome data with age labels and you use relatively simple machine learning, it's not like fancy transformer models or graph neural nets, just simple linear models. You can train a biomarker that's highly correlated across multiple tissues. So that's the first narrow AI. So there with the original clock, we've gone one step further, which is a single cell aging clock that basically brings the epigenetic clock into the single cell world via gene expression. So there's a bit of complexity here, which is you've got the epigenetic clock, which can hear aging. Doesn't tell us what aging is, but can hear aging, right? You've got all these methylome sites changing, and it gives you like the sense of aging. And then what we've done is from the gene expression or the gene signature, we can hear the epigenetic clock. So the epigenetic clock hears aging, and we hear the epigenetic clock from gene expression. So it's, for lack of a better expression, it's like Chinese whispers. So, epigenetic clock hears aging, and we hear epigenetic clock, and the message might be slightly distorted, but it does give us high throughput exploration on the aging problem, right? You're able to do like single cell CRISPR screens and see which genes are affecting aging. Um, and that brings me on to the second narrow AI tool that we've developed. So this isn't our sole development. This is something we've fine-tuned from the literature. So there's these open source virtual cell models. So I'm going to give you a very precise definition of what virtual cell means to us. What it means is a representation of how genes impact each other. And the way that this is created is you feed quite advanced machine learning models now, like transformers and graph neural nets. You feed them loads of single cell gene expression data. So these models look at lots and lots of cells, they look at what the genes are doing, and they basically stitch together if gene A is up, gene B is down. If gene D is up, gene F and Z are down. So it can, from the ground up, establish how genes interact with each other. You can put that in a computer, And now you can effectively run virtual experiments. So I can say I'm going to overexpress 2 genes or 4 genes like the Yamanaka factors. What does that do to the rest of the genes in the genome? And because we have another narrow AI tool, which is a single cell aging clock, we can convert what's happened in that in silico experiment into an age label. So we can say what was the age at the beginning, what was the age at the end, and so we can say, look, what was rejuvenative out of, were these 2 genes rejuvenative when we overexpressed them? Were these 4 genes rejuvenative? Like how more rejuvenative was one experiment versus the other? So you can see we can do a lot of stuff all of a sudden. And I think just to try and emphasize how powerful that this is, the number of experiments we can do within a year is roughly 3 centuries of future wet lab experiments. So it's like getting into your DeLorean, going to 2325, getting out the DeLorean, like reading the results, right, like the best results, coming back to now, right, and doing that in the wet lab. And that's sort of the power of like having things in the computer, having these aging biomarkers you can use in these types of situations. So these are 2 narrow AI tools, right? Single cell aging clock, virtual cell. We use one inside the other. I call it like the dance of the black boxes. Um, and then, yeah, just very, very long answer, sorry. But then a
No.
last AI tool, and this is something we're not deploying right now, but it's this idea of the AI taking over from the scientist, like sort of taking over decision making, deciding which experiments to do. I think it's like agentic AI, right? Like sort of making a better scientist, like an AI scientist. So I think we're not quite there yet in terms of like, you know, we're 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 at the mercy of that decision maker, but it's certainly getting more powerful every day. And this is where, when people talk about AGI and superintelligence, imagine that in combination with these narrow AI tools, like the amount of, iteration that's going to be happening relatively quickly. So, we're already finding things with our mushy biological brains in the mix with the narrow AI. So at some point, I don't know when that is, we prepare for that. We basically prepare as if that's never going to happen. But if that does happen, that's going to be a huge supercharger as well.
Well, yeah, I remember the quote that some of the people are making now about scientific discovery powered by AI, and some are even predicting that, in the next 10 years, we're going to have more scientific discovery than we've had in the last 100 years, based on the power of AI to drive innovation and discovery. And since you mentioned that Steve Horvath was at UCLA, which is just down the street from where I'm sitting here today, when he did the original methylation clocks back in the day. Now he's up in Altos in the Bay Area, but that work there now. Where does that fit into? I assume you're not using LLMs or even deep convolutional neural networks. Is it deep learning at all, or is it more, what type of AI are you using on these?
So the answer is all of the types. So we basically we have all the data, and then we try all of the different models, and we basically just look at the result, like which model creates the best single cell aging clock. And I think the answer I can give you is it's actually a mix of a lot of things, right? It's like you need to be a good chef, right? You need to try a lot of things, right? There's obviously lots of things that aren't going to work, but we mix some of the models together. That's also true for the virtual cell. So I guess one of the models we hold in high regard is called SCGPT. So it's called single cell GPT, and that's a transformer model that sits underneath. There's a graphed neural net version of the virtual cell. I think it's called Gears, and there's a more recent one called State. So there's all sorts of models, and we just stay agnostic to model. We just try all of the models and see which ones are most accurate, which ones are most robust. Um, so yeah, it's a sort of a strange process, right? Because you just try everything and then you choose the best thing. It's almost like how evolution operates. It's not thinking, right? You're thinking enough, right? You're like, here's a bunch of variety that we want to test, and you're gonna source that, but then ultimately you're just gonna see like Hunger Games, who lives, who dies, or it's more of a hierarchy, right, versus life and death. Um, but yes, there's not that much putting your brain in the mix apart from just trying to steer this in the right way, keeping your eyes open. Yeah, I think that's, did that answer the question, Robert?
Yeah, yeah. I mean, it's almost it's what we're hearing more and more with sort of the modern deep learning paradigm where we don't really program the computer with the task. We just program it with the tools, the convolutional neural network, the LLM, and then it learns from the data, and the data is domain specific. So to some extent, you don't even have to. I mean, obviously, you guys are domain experts and you're adding a lot of value there, but a lot of it is the machine. It's fascinating. I'm thinking of. Go ahead. Yeah.
Yeah, so I think the definition of expert has changed very quickly. So in the world of machine learning, expert means the data set that you have. So, did you build an expansive data set with high quality control on the data? Because that's going to give you the expertise. If you have a messy data, if you have a messy data set, poor quality data set, you can't be an expert with all of that noise. So, yeah, machine learning, like the data set conquers all. With a great data set, you can actually use pretty primitive machine learning to reach your outcome. But then if you have a great data set and a great model, right, you can hit new heights. If you have a bad data set, it doesn't matter how good the model is on top of that, you're just going to be hitting false positives and negatives. You're going to be, you're going to be stuck in the noise. So yeah, that's I guess our definition of expert is not a human definition anymore. It's a sort of machine learning definition, which is like the data set. I think, there's many examples of this. So BioAge, like where's their expertise come from? It's this big data set that sits underneath, right? They have this sort of biobank data that they have exclusive license to, and many of these sort of really exciting new machine learning in biology companies, it's all about building these huge data sets. So yeah, the human brain is out of the mix, right? It's just sitting there in the data, and the machine learning can see things that are just like many levels below the surface.
Yeah, and it brings me to kind of your third step there. It reminds me of some work. George Church is a friend, and he's a geneticist based out of Harvard. He famously founded Colossal Labs, to bring back the woolly mammoth and but he's done a lot of other stuff. But one of the things he's working on right now that is amazing. Is the Lila Labs, which is a dark lab that does AI generated hypothesis, AI generated testing, and then even the AI driven robots that do the pipetting and the smearing and the feeding the mice. And it's called a dark lab because there are no lights, because there are no humans, and it runs 24 hours a day continuously testing and innovating and rehypothesizing kind of what you're doing. And they've just raised $500 million for this project. And they're one of several dark labs that are going this way. There's even a dark automobile maker in
Yeah.
China. That's the dark factory. I'm blanking on the name, but I'm sure there'll be more. But and that's sort of your next phase, or you're already doing heavy AI automation. It sounds like you're doing it with the hypothesis testing. Are you doing it with the actual wet lab part as well? Have you gotten there yet?
Yeah, so these sort of dark labs where you have the whole scientific process, that's how you, like, how do you iterate as fast as possible, right? It's those sort of setups. It's like I'm going to test something, and then I'm going to see what the I've got an idea, what does the real world say? And then I'm going to use that information to feed, like, update my model and just keep going around until distance between real world and my model is minimum. Um, we've got our setup slightly different. So it's more that we just do every experiment. So there is iteration, but it's actually relatively infrequent. We just build tools, we do every experiment, we end up, enriching the signal, right? You just take the extremes of the distribution, and we take that into the real world, and there may be some error, and then we can use that to update our models. But it's mostly just throughput. When you put the laboratory in the computer, so this is not a this is, I mean, inside the computer is dark as well. Maybe there's a few sparks, right,
Right.
as the electrons move around. But like, I think that the better description of what we're doing is like the whole lab's in the computer. Like we're just doing every experiment in the computer and just taking the best things to the real world, and then we can update our systems once in a while, right, based on all that data that we generate in the wet lab. But the great irony of our wet lab is that most of the wet lab scientists are just generating the data set to make a better model. So, most of the expense of the company is simply data set and managing that properly. And then occasionally we get these predictions from, this machine learning in the cloud lab, and we bring that home and the scientists will test that, and we're getting great conversion rates. So yeah, we feed this model and then we see what it does and digests, and then it sort of throws us a crumb, which is this prediction, and we test that. um, so it's slightly different to these dark labs, right? It's not quite as sci-fi. But actually I'd argue it's sort of more
It's more
sci-fi. It's just,
sci-fi.
yeah, yeah.
Yeah. Sorry, didn't mean to interrupt. Yeah.
But it works. That's the thing. It actually works. So, it sort of sounds exciting, but if it doesn't have utility, like it doesn't matter if it's exciting, but it actually works. Like that's the amazing thing. So, all of these tools were, we were trying them for the first time. We didn't even know if they would do anything useful, but we sort of put them together and they did something useful. And now you can iterate, and we've got much more powerful tools. And the question now for us is, did we harvest everything the first time around? Is there anything left? The second time, right, if we gear up with better tools, are we going to find as much new biology again, or are we only going to find like an increment, like an extra 20%? So yeah, that's our next step. Like when we turn the handle of the machine the second time, like what's new and what's already been seen, and that's going to give us an idea of like, have we harvested everything, and now is it time to move on, like fully double down on progressing biology to different sort of therapeutics.
Yeah, yeah. Well, pivoting now to Shift Biosciences, and particularly what you're doing with the Yamanaka factor driven partial epigenetic reprogramming, right? When Shinji Yamanaka did that, he had these transcription factors that activated a whole bunch of genes that are presumably related to embryogenesis, right? To the natural thing that happens when our cells reprogram our epigenome, which happens every generation, right? Otherwise, we wouldn't be there. So it's somehow tied to that. Now, your approach is, I mean, it's different in several ways, but you're identifying individual genes that either accelerate aging or slow down aging, right? So much fewer genes. Do you see yourself? I mean, is it is an advantage, I guess, computationally, and you can answer that. But do you see yourself as doing like a symphony of genes, kind of like the Yamanaka factors do, sort of a polygenic effect? I mean, is there a disadvantage if you have to do it gene by gene by gene versus the good news is you know exactly what genes are, whereas the Yamanaka factors you don't necessarily know, correct?
Yeah, so we expected when we tried to search for, things that weren't Yamanaka factors, that there would be another combination. Yeah, that was our assumption, right? There are four Yamanaka factors. Maybe you can get away with three Yamanaka factors. But if we're going to find anything better, it's also going to look like that, right? Like that makes sense. Like what do we know about rejuvenation? Just Yamanaka factors. So you don't want to stray too far from far from home. So we geared up to explore combinations, right? That was having a single cell aging clock and having a virtual cell, so we can explore combinatorial space. So just for just an example, if we want to test all combinations of three transcription factors, which is sort of the minimal subset of the Yamanaka factors that is rejuvenative, that's 562 million experiments. And we are talking like centuries of wet lab experiments to do that. And so that necessitates having a virtual cell sort of, run a meaningful exploration, right? If you only sort of, it's like battleship, right? If you only put a few pegs down, you're never going to hit the ship. And if the
Yeah.
ship is 562 million holes, and
Yeah.
you only got three shots, yeah, it's like, it's sort of impossible. So, so with these tools, we can actually put a peg in every single hole. That's the amazing thing. So this is the thing. We geared up for combinations. The first thing we discovered was there were single genes that were common to combinations predicted to be rejuvenative by the virtual cell. So this gene kept cropping up. And like we're just looking at it, like because we still get to look at the results, right? As you know, we don't abandon the science entirely. Well, we just look at things and just what you can notice. This is like basic recognition, right? Oh, there's a gene, and it's coming up multiple times. And so we didn't believe it could be that easy, right? That here's 3 to 4 Yamanaka factors, and now here's a single gene that's potentially driving all these phenomena. And so we just speculatively took it into the wet lab. We actually just went to the gold standard, for us, epigenetic clock assays immediately. They're quite low throughput, even though it's better than lifespan, it's still low throughput. And huge surprise, this single gene rejuvenated multiple cell types. It rejuvenated more than Yamanaka factors, roughly twice the velocity of rejuvenation, and no pluripotency. So yeah, we were doing like a double take. We are like, no, single gene. Like, let's try it again. It's the same result. Let's try all the clocks, right? Maybe it was the first clock, and then we said, no, it works across all the clocks. Do the pluripotency assay again. And actually, when you think about it, like your assumptions going in, they color your conclusions. So yeah, people were like, it's not possible. But then the only thing we knew about rejuvenation beforehand was Yamanaka factors. So like, but the definition of rejuvenation was pluripotency. So like, you shouldn't be that surprised that if you directly optimize for rejuvenation, you don't necessarily bring across an entirely different pathway that changes cell identity. So um, the irony of this story is that we geared up for combinations. We built all of these machine learning tools, and the first thing it told us was there are single genes where we don't even need these tools anymore. You can just systematically go through single genes in the wet lab without a virtual cell, and that's what we did as a second step. And then the big surprise was how many single genes are influencing the aging process. So we ran a poll in the company, right? Like everybody had to guess, and whoever was closest won the prize. I don't remember what the prize was. Something very modest, yeah. And I think I put down 10. I was like 10 out of the 1,500 genes we're going to test will be involved in aging. Some crazy guy put 150. He wasn't crazy enough. It was 190 genes. So it was like roughly 10% of these 1,500 genes were impacting age. About 150 was in the rejuvenative direction, so the majority of the 190, but 40 were in the pro-aging direction. And amongst that, we saw Yamanaka factors, the combination of the factors, because we wanted to use that as a control. The individual factors didn't actually feature very highly. But then we find all these single genes, that some of which outperform the combination of factors. And this was a huge surprise, just the sheer volume of biology impacting these processes. And then, just the fact they're single genes. So, yeah, I guess the funnel, right? Virtual cell, I think there was like 350,000 combinations of two. Then we took 1,500 into the wet lab. From that 1,500, we found 190 with a relationship. We took the top 50 from that broad screen into epigenetic clock assays, and about 30 of those genes reverse epigenetic aging, 10 of which outperform Yamanaka factors. So you can sort of see the numbers. Um, and just last point on this single gene that we discovered initially in the simulation, it's actually part of a family. So the transcription factor family, multiple family members are rejuvenative, but one of the family members has no rejuvenation. So you've got very similar protein sequences, and then you've got this one member, again a similar protein sequence, but no rejuvenation whatsoever. So it gives us this sort of window into mechanism, because you can find what's the specific sequence on the rejuvenating family members that's absent in the non-rejuvenating family member, and you can start like co-folding other proteins to that sequence, and that suggests other candidates that might be mediating rejuvenation downstream of the transcription factors. So yeah, this gene was the most rejuvenative, but then it revealed a family. So it's not just some random gene, right? There seems to be a cluster of rejuvenation genes. So it feels to me like we're getting closer to pathways and mechanism and things like that. But yeah, just having the tools to do a broader screen, optimizing for rejuvenation, the amount of power you now have on the problem with that little bit of extra information is, sort of, it's the whole point of science, right? Like, how do you get there, right? You get better information along the way. That creates a new experiment, or that creates the need for a new tool. Your information is sort of ratcheting up, and compounding, and then you eventually sort of burst into this new landscape, and you just, it's something to behold. We're not quite there yet, but you can sort of see the outlines of this happening at the moment.
So that, I mean, that's sort of a holy grail almost, with the rejuvenation of the Yamanaka factors without the downside of the pluripotency, and even greater rejuvenation effect than
Yeah.
the Yamanaka factors, right? Yeah.
Yeah, I mean, there's plenty of ways this can still fail, right, as we move
Yeah.
forwards. So, we do, there's boundaries of our assay. So we do a colony formation assay in vitro. We express these genes constitutively, so continuously for 6 to 8 weeks. So up till 8 weeks, we're not seeing colonies for these single genes. We see them for OSK, we see them for OSKM. Maybe you pushed it another 2 weeks, they'd start to emerge. I don't know. Maybe we're going to try this we're going to try this gene in an animal. We're just going to make sure, like, does it, is it safe in the animal? But um, certainly, if you just compare what's known, like Yamanaka factors, to what we're finding in the early assays, there's a lot of new stuff that's cause for excitement. So there's certainly these scenarios, right, where some really interesting things could happen down the line.
So I have to ask a question, and to someone who's not really hypothesis driven, this may be a completely inappropriate question to ask, but I wonder what the teleology, or the biological use for these genes that you've identified. I mean, obviously the Yamanaka factors are targeting a embryologic reset, the sort of a natural mechanism. Are the genes you've identified, do you think these are just a subset of or maybe a superset of the Yamanaka factor genes? Or what are these genes for? Why do we even have them in there? Or is it not appropriate to think of teleology, just like we don't need to think of hypothesis? All we care about is results, right?
So I think there's a healthy level of hypothesis, which is, if you just try to join the dots, it might reveal something obvious. Like, I think there's always a case, right? If the dots are in front of you and it really looks like a duck, right, without joining the dots, and then and that tells you about what you're dealing with. So just a basic question we ask, which is like, where is this gene normally expressed in the human body, and when is it normally expressed? Um, it's expressed in very early development. I believe it's around the time of the natural rejuvenation event, the sort of post-conception. I think it's the first few weeks, at least in mice. So that's very provocative, right? The fact it's, you know, in terms of the timing, it's sort of expressed around that time, and then it's sort of shut off for the rest of your life. And it's under very strict control. So when we try and overexpress this gene, the biology of the cell is like trying to shut this thing down. It's like methylating the endogenous gene, not the exogenous gene that we're introducing, but it's like absolutely methylating, trying to deregulate, downregulate this thing. So that's really interesting. I mean, all transcription factors are very influential proteins, but the fact this one is under such strict control is very interesting. I think there's also this association with the germline, so not really expressed across the body, but also in the germline. So again, that's quite provocative. Um, but we don't know if this is just correlation or causation, right? Like, it sort of feels like it could be elegant, right? Like maybe this is some of the biology that's involved. But again, we just want to do a comprehensive survey and then like validate everything to the the greatest depth, and then see what survives, see what sort of turn out to be a false positive from our screening system. And then I think once you've got really high confidence in the biology you're looking at, that's the best time to try and piece together a story.
When you spoke earlier about mitochondria and the evolutionary migration of the majority of the mitochondrial genome to the nucleus, it went to chromosome 1. Um, is that correct? Yeah, I thought it was
Y-
1.
I I I don't. It's a long time ago, so I hung up my mitochondrial boots around 2018. So it's not that long ago, but um, I don't remember that level of detail. I'm impressed that you got chromosome
Well,
numbers in here.
I never did mitochondrial research at Cambridge, so I shouldn't shouldn't speak about it. But whatever gene, my recollection is localized to one gene, and even Aubrey was talking about moving some of the mitochondrial DNA, which is easily damaged in the reactive oxygen in the and move it back onto the chromosome, and even move it that way to get less mitochondrial DNA damage. But I'm wondering, I wonder if any of these genes that you've identified come from the chromosome where the mitochondrial DNA is located, that they could possibly be, at some level mitochondria DNA as well. I hate to keep kicking it back to mitochondria. I was just curious. Yeah.
So yeah, so I mean that is an interesting idea, but what I would say, what I can say is that these genes we tested, vast majority were transcription factors, and the reason
Okay.
was,
Mm.
where did we start? They were Yamanaka factors, like that was our definition, and they were all transcription factors. So inherently, the broader screen, we were like looking for alternatives. So we started with transcription factors. There was some biology that wasn't, it was beyond transcription factors. I don't recall whether there were mitochondrial genes amongst that. So I can't confirm or deny, right, whether this is true or not. The second thing, just to speak to mitochondria, I think this is a question that deserves a bit of time, which is like, what doesn't epigenetic repro like what are the things that epigenetic reprogramming can't do? And there's actually quite a large laundry list, and it's not in my interest to talk about this list, but I think it is necessary to sort of, you know, appreciate the bigger picture in biology. So there are two genomes, okay? So there's a nuclear genome and there's a mitochondrial genome. So there's basically two operating systems that have to cooperate together. So of course they're both important. Like of course, it's not like one is like unimportant and one is important. So epigenetic reprogramming is principally affecting the nucleus. So when we reprogram the epigenome, basically taking gene expression in the nucleus back to a young state, that does not reset mutations in the nuclear genome. So we can't undo damage. So like we can't go back to the blueprinted genome. We don't reset any issues in the mitochondria, at least not at the genome level. So we can't affect mutations that are potentially taking over. Um, and yeah, there's many other things like we don't reverse cells that have already died in the body. So there's plenty of parts of your body where you're losing cells, you're not replenishing them sufficiently, like dopaminergic neurons in in your brain. Even healthy people lose them. So that's another thing that reprogramming doesn't reach. Autoimmunity. So if you start developing autoimmunity, those antibodies are there. Those white blood cells have created those antibodies. You can't, you make them younger, you might actually make it worse, right? They're just producing more autoantibodies. So there's like a whole car crash of stuff that can happen that is potentially, well, many of it is irreversible. Um, so cell reprogramming relies on the cell still being alive, not too much damage being in there, the mitochondrial genome still being like some sort of fit for use purpose, fit for use state. So it's by no means a perfect therapy. So I'm quite uh, fond of mitochondria because that's where I came from. I really believe that was a big piece of the picture. And from these transfusion experiments, it continues to sort of show quite dramatic results in certain use cases. So I think both genomes is going to be an increasing theme, like epigenetic reprogramming around the nucleus, like epigenome, and then mitochondrial transfusions, right, like deal with the mitochondrial genome and have more energy producing organelles. So yeah, getting into this like silver bullet, golden bullet mindset is basically betraying biology, right? Biology is there's minimal units in biology, and the fact we got two genomes is like just the basics of the cell.
Yeah, that's such a good point to keep that in mind. Now let's talk about the good stuff. We could speculate. I mean, that's all good stuff. This is all super exciting. But going towards the whole idea we were talking offline about, that we're entering a new era of potential therapies for the age-related diseases that now take up 80% of our healthcare dollars by some estimates. These chronic diseases of aging, we're now seeing the idea that we can approach them with rejuvenation, and somehow these rejuvenation strategies will work on possibly all these diseases of aging, assuming there's not like permanent damage, like you mentioned, on, nuclear DNA or other, the immune system as well. But it's a fascinating idea of doing rejuvenation as a strategy for chronic disease of aging repair. So with that long-winded introduction, what are your targets? I mean, you guys are away from clinical trials as well on this. What do you think? What's the low hanging fruit for what you've developed here?
So, sort of a two-part answer. So we have crystallized these two targets. So one is a expression target, like Yamanaka factors. You overexpress them to rejuvenate a cell. So this is a single gene we express, so somewhat like the Yamanaka factors. But to our surprise, we also have another target, which is an inhibition target. So we can knock down this target uh, right now by siRNA, but we might also be able to do this potentially by small molecule to rejuvenate the cell. And that was very unexpected, right? To be able to inhibit a gene to rejuvenate. The way we found this gene was indirectly when we did this screen of 1,500 genes, most of which were transcription factors. Before I said we found 150 genes that were rejuvenative and 40 that were pro-aging. Now, one of those pro-aging genes was very special because it was widely expressed across the whole body. And that's exciting to me because I want eventually the whole body to be rejuvenated. So to inhibit a gene, it needs to be expressed in the first place, right? So it sounds obvious, right? But you need the gene to be expressed if you're going to get in there and inhibit with a drug. So this special gene was expressed across the body. Potentially we could inhibit to do something beneficial across the body. And when we looked at this gene, in the literature, it had been linked to lifespan. Inhibition of this gene had been linked to lifespan, and inhibition had been linked to fibrosis. So the fibrosis was really exciting because it was the first time the data took us to a disease. It's not like here's all these age-linked diseases, which one do we choose? Go through all the variables. Like I sort of do a Rain Man right on the problem space. But this was no, let's do a screen. Let's see where the biology leads us, and it led us to fibrosis. So we basically did our own assays because there's a reproducibility crisis. You don't want to be caught up in that. Do your own assays, your own systems that you trust, and we knocked down this gene. We rejuvenated, and then we prevented fibrosis by using this approach. So these are two different sort of two different sides. So overexpress to rejuvenate, and then knock down to rejuvenate. And clearly that one's tied to fibrosis. Okay, so that's there's a case there to be made, and there's every organ across the body tends to have a fibrotic form in a disease. Like in the liver, it's called MASH, diet-induced fibrosis. You've got chronic kidney disease, you have fibrosis, you get heart fibrosis. So you can imagine, right? Like you could develop a drug, even just to a single organ, but for the fibrotic form, and then you could do that for a different organ, like for that fibrotic form, and you sort of got access to most of the body through the fibrotic forms. Um, so it creates an economy to develop something. On the overexpression side, there seems to be a very close relationship between aging and the anatomy of the inner ear. So with hearing loss, the anatomy changes in a very, well, not linear way, but in a very predictable way that maps very closely to aging to the extent you can almost use the inner ear as an aging clock. Like it's so tied to age. So we're actually using that as a use case for expression because the anatomy is tied very closely to aging. And secondly, you can actually get genes to the inner ear by local injection of a virus. So not huge amounts of material that causes an inflammatory reaction, but just a small amount in a site that has partial immune privilege. And we might be able to do something really interesting for a disease that has huge clinical need, right? You might know people that have hearing loss. I certainly do, but it's like a big thing out there that doesn't have a therapeutic. It's just hearing aids. So yeah, we've got hearing loss on that side, fibrosis on the other side. In terms of translation, I think the best description I've got of it, like the more you realize about drug development, the larger it looks. It's like a mountain on the distance. You're like, oh, that's a small mountain. And then as you get closer, you're like, oh, that's a bigger mountain. And then you get closer and it's an even bigger mountain. You just keep approaching it. It just gets bigger and bigger and you're still far away from it. And it's absolutely astonishing the amount of work that goes into making these drugs real. So I'm not, I'm not completely naive to it, but it every day it continues to amaze me how little I know about drug development. Like the more I know, the less I know, and how much effort it's going to take to just, struggle to move these things forward. So, um, it's great that we're finding these things in the lab and we're sort of crystallizing around indications, but like that's just the beginning, right? Like the amount of risk that needs to be managed, um, progressing these things is enormous. So I think, yeah, it's almost like a perpetual learning experience, right? As we move forwards, it's like, oh, you know, a new chapter has arrived, and you've got to understand this, and it becomes engineering. But I think it's important to emphasize just how large and multifactorial the problem solving space is just for drug development, let alone, you know, I've got biology that's validated. Just the journey ahead is very hard. So there could be like a huge amount of promising stuff at the start line, but the finishing line is going to be complete poverty. Like just some poor runner that's just collapsing over the finishing line. Like that's almost the, you know, it's just the amount of trials ahead of us is enormous. So I don't want to put a dampener on it, but I think we should get comfortable with the idea that this is a big challenge. We should gear up for it. We should like get our temperament in the right place. Like we're just gonna have to keep going at this. It might take longer than we expect. We should be creative, but we shouldn't like put patients at risk. So I think it's just like sizing up this mountain is an essential activity because otherwise we'll just end up being hopeful and disappointed and we get jaded and you know we just abandon it and then we come back later because that's really what we were excited about and you know it all becomes very disruptive. But like we size this thing up appropriately, we can just you know do the work that needs to be done. And it's like this expression, right? The shortcut is the long way. You know, it's like just you just need to do the work that's necessary. So that's, I guess, that's something that's more new to me, just the size of the drug development challenge. But it doesn't put a dampener on it for me. But it's something that I think needs to be appreciated like sooner rather than later.
Great points. So much to unpack there. Let me hit on a couple of those. The hearing loss is that's great with the age-related hearing loss. Certainly, I mean, even rapamycin. There's animal work showing that rapamycin reverses presbycusis, which is the age-related hearing
Yep.
loss, and the anatomy that changes. Is it particularly the cochlear hair cells, or what is it? Do you recall what it is, particularly in the anatomy and the cochlea, that changes with the aging? I think that's what they were counting with the rapamycin studies. It was actually cochlear hair cells. They could measure those, so it may be that. I'm not. Yeah,
Yeah, so again, I guess like we're taking an agnostic approach. So there's a lot of
yeah.
evidence the inner ear hair cells are, you know, involved, right? And there's clearly a loss of these inner ear hair cells that become damaged. But there's also changes in the broader vestibular compartment, which is sort of the rest of the inner ear. So when we go into mice for the first time, we're gonna go into as much of that anatomy as possible. So the inner ear hair cells and the vestibular compartments. We're going to try and deliver a rejuvenation gene across the board just because we don't want to be presumptuous. We don't want to second guess it's just the inner ear hair cells. Because what if it isn't, right? What if you need
Right.
to do both? So we're going to go in broad and then if it works, then you can say, okay, let's narrow it down. Right, let's just get to the inner ear hair cells, and is that enough? And then okay, that's enough, and then we can be more efficient. But again, we don't know for sure. We don't know for sure, and we don't want to second guess. So we just try and stay agnostic. So again, it's sort of unsatisfying to hypothesis-driven science.
No, there's a beauty to that approach. Forgive me, I was falling back to my old ways of hypothesis driven, but I like your approach, and I think that's a great way to do it, and it's obviously been successful for you. The challenges with the animal work versus the in silico AI work, it's you can do hundreds of thousands of experiments in a short amount of time with silicon, but do you see are you able to bring AI to the animal work? I mean, we talked about the dark labs and that kind of thing. Do you see AI accelerating that animal work, or is it basically now you've hit the usual thing and it's going to be years of work with that?
So one day, it's not today, and outside of specific use cases, but one day we'll have the interventions that we found, like in the virtual cell. We'll put them into the cells in vitro in a dish. We'll put them into the animal. Some things will work, some things won't work. We'll then progress that, right? We'll progress it down the whole stack of development, and we'll be able to train a model across the whole thing. And the more targets we progress from start to finish. It doesn't matter if they're failures or their success, although obviously the successes get further, right? So you have to be quite disciplined. But the more we progress, the more powerful the model we have that we can train across the whole thing. So there will be a point, it's gonna require a lot of investment against a landscape where it's hard to raise money for drug development, like let alone make the experiment broader, right, to generate more data. But eventually there'll be a model where at the very earliest stages, based on the data beforehand, you can predict whether that intervention is going to be a clinical trial success. Obviously, there's going to be error bars, right, and there's going to be a certain confidence, but there will be a point, and these models are being trained right now. There will be a point where at the earliest stage you can predict if this is going to be a clinical trial success in humans. So the use case that has been built is for liver toxicity. So um, basically, there's a lot of data for molecules that have worked and haven't worked between the earliest stages and liver toxicity in humans, right? There's sort of a critical mass, and a model's been trained across that. And so you can actually, at the very earliest stages, assay for liver toxicity, like basically in a simulation, and avoid that issue, which is one of the big causes of clinical trial failure, right? Something gets liver toxicity. So like Bioage, right? Their intervention, it was a liver toxicity issue. So this is, that's one use case. But eventually there'll be a model that crosses the whole thing, and you'll be able to say, is this going to be a gene that can affect lifespan? Obviously, health span and lifespan, right? That's my wish, right? Health span and lifespan together. But you'll be able to sort, in the future, I don't know how long this will take, how much investment, but you'll be able to say, like, will this affect health span and lifespan in human beings with certain amount of confidence? That's pretty cool.
Yeah, yeah, this is so exciting. I want to be respectful of your time. Just maybe a couple more points if you can. We talked
Yep.
offline about the FDA and the challenges of a clinical trial, the expense, and the theme of this conversation. A lot of it has been don't use hypothesis or well to step away from hypothesis driven ideas and be open to what the data shows us, and with AI that's possible and all. Well, the FDA is almost the antithesis of this. They are asking for a definite hypothesis, and so it really limits your choices, and it's really it's expensive, it takes time. So what do you think about the role of citizen scientists about expanding, testing with drugs? Obviously, it'd all be done within a legal framework, of course, but the idea to, once safety is established, at least open up to, many possible chronic diseases.
Great question. So the nature of what we're doing is very expensive. So this sort of target discovery for cell rejuvenation genes is some of the most expensive basic science you can imagine. Like we eat single cell omics reagents for breakfast, lunch, and dinner, and these are like some of the most expensive experiments like you could do in an academic lab. So the burn rate, right, is just sort of makes my eyes water sometimes. And where do you find the money to do those types of experiments? It needs to come from meaningful capital pools. So, we have the initial check and shift was relatively small, but then eventually you have to enter VC territory, and the critical mass of capital is like biotech VC, right? And they want this to live in biotech, right? That's where the capital is, and that's you have to develop something there. And then you get to the situation where like there's these timelines, and there's all the there's regulatory checkpoints, and there's sequential financings you have to get through, and you have to sort of please a lot of people. It becomes this sort of this thing that you have to manage, right? That's very different from the basic science. Um, like, do you continue to plug away, right? Like just struggling on, just putting up with it, or do you like get out of that system, right, and try and do something on a more aggressive timeline, sort of manage the risk yourself, like just try and create something in a different system. As a developer of drugs with backing from biopharma, my best answer at the moment is we have to build it in biopharma because if we build it in biopharma, it can cut across to the other side. If we decide to do something over here instead of biopharma, it's very hard to cut across back to biopharma, and I sort of see biopharma as a distribution system. If we build it here, It's going to go everywhere very quickly. If we build it over here, it's going to be in the long run, it's going to be harder to distribute. So I think I think we have to build it in biopharma. Like that's not optional. I don't think we can abandon that. I think there's processes that are slower than they need to be. But also, there's a lot of really great people there and checks and balances that are there for very good reasons. The idea that as a startup company, I'm going to reinvent that whole system on top of what I'm doing, which is this enormous, mountain that needs to be climbed. I think, we're thinly spread already, let alone reinventing the whole drug development system. So I think we have to build something there. But I also recognize that there's people that they're in a lot of pain today, right? Or they're running out of time, like they've been given 2 years or something like that. They can't wait for this thing to take 10 years, 20 years, right? In its fully flourishing form. So I think there should be a button, somebody that can press before cryopreservation. There should be a button someone can press, which is like, I'm out of options. And there's this amazing technology today. There's sort of I guess, surveillance of AI. Like when you're doing new things, right, you can be more careful about what's going on and shut it down. So you've got all this amazing technology, and I'm not able to press a button and use the best that's available ahead of, so I think there's like compassionate use. There's sort of different versions of this, but I think that's something that should be formalized. I think just saying, it is the way it is, right? You've got 2 years, there's nothing we can do. You should go to a hospice, enjoy the time that's left. I sort of feel like, I think there should be like a courageous hospice. I'm not saying I don't want to frame people, because that's a really difficult situation. There's a lot of courage in there, but I think there should be a sort of a proactive hospice where people are like, well, I'm gonna try something, right? And I'm out of options, and I'm willing to try something when otherwise it wouldn't be tried. So I think it'd be very helpful to formalize something there, um, and sort of build that into the other system. And I think that's going to take a lot of people, right, to sort of create that. That's not something I could do as a side project, right? That's an undertaking in its own right, but I think there's appetite out there. That's why I appreciate sort of Tom Benson and what he's done with the
Yeah,
Nightingales, right? It's like a proactive approach to your situation instead of just making do with your lot. Yeah.
yeah, and we talked a little bit offline about that. Yeah, but I'm working with a group that in the United States, many of the states are starting to do these so-called right to try laws. And originally it was for people who only had 6 months to live, compassionate use. But now at least one state, Montana, has dropped that. So any, adult of sound mind can take drugs. Right now, they still have to have passed phase 1 clinical trials with the FDA, but even that may be rolled back because, as adults, we should be able to, make choices about what we do with our bodies. At least, well, I don't want to get political or anything, but there's a growing sense of that in the U.S. and there are places now that has been codified. The regulations allow it, and so I think we're going to see more and more of that. And if the regulatory framework is changing, so if we now just need kind of a good scientific framework, so we get all the data, we don't lose the data, and just have a bunch of people in the Wild West trying it. But
I, I think like a version of this, I don't know exactly what it's going to look like, but if there was some AI or narrow medical AI looking over your shoulder, like plugged into you whilst you're trying these things, like trying to make this as safe as it can be, I think that would be very helpful. Some like massive scrutiny device that has it's got machine learning's built into it, helping people navigate the front lines of medicine, right? Like being the n=1 for the first time. Don't hit this landmine, right? This thing can give you an early warning system ahead of any human seeing that. Like I feel like that's a very promising direction. Like, let's make the most of today and these tools that are becoming available to allow people to explore, where previously it was huge amount of sort of treacherous risk. Um, but yeah, it'd be really nice to. I don't know exactly how this is going to get formalized, but it just feels like something that should be built.
yeah, yeah. I mean, it's definitely coming. It's already happening. So hopefully that will come sooner rather than later. And This has been great. Anything we didn't cover, Daniel, today that you want to end with, or this has been such a great conversation.
Um, I think just there was one question I put on my top 5 questions I'd like to be asked,
Okay.
and it's when did you get closest to giving up? So I sort of described this
Yeah.
this experience of seeing a mountain, really like covering a lot of distance, but the mountain's still the same size, and then you're like, oh, it's bigger, and then you keep going, it's like, oh, it's bigger. So I think a perfectly rational response is, when is it going to get bigger? Like I'm playing the long game here, but if winning is on the other side of death, right? It's not really a game, it's like I'm just, yeah. So like when did I get closest to giving up? So there's a few examples of this where I think I'd been in cell culture for like 3 or 4 months, and I should have only been there 2 months because you know that was the duration of the experiment. But like about 2 days from the end, all my cells got contaminated, right? This is something that happens in cell culture. To reset the experiment, it's like 2 months down the drain. So like you set it up again, and then you're like really, you're overly policing the area. You're being quite mean to people because you suspect everybody was. So you can just see like this, your character's getting, you're getting in a bad place, right? And it's not, you're sort of miserable but I sort of, it was like, if this result doesn't come in, and I remember there was this instrument called the pyrosequencer. Like I knew what the shape of the graph would look like if we'd reduced mutations in the mitochondrial DNA. My heart was beating. 'Cause it's like 4 months of work, right? Was it all for nothing? And it came in as absolute like euphoria, right? Like you sort of struggle, like you haven't got any melanin, right? Because you've just been in like a bunker in cell culture. It was like, if this thing doesn't come in, that's it, right? This is not, this is crazy. Like this situation's crazy. But then it came in and it was absolutely worth it. And many times I've been in these situations where, you see, your friends like moving on in life. There's things you want in life that aren't just solve aging, right? There's like other things. You see people moving on. And like meanwhile, you're just stuck in cell culture and they're like ringing you like, oh, we're having a great time. Where are you? And you're like, oh, I'm with my cells. And they're like, not again. But yeah, but but each time I've had a number of these, right, when if it doesn't work, I walk away. But it's always worked. But it's a you know you worry that it's only going to fail later. Like the damage is only going to be larger because you'd never know for sure, right? It could be like this initial hypothesis, right? Like I believed I'd solved aging and I was waiting for this signal, but that wasn't actually solving aging. So you always worry that now you've wasted more time, you have to go back to square one. But there is a point where you've got sufficient breadth on what you're doing where there's like 5 shots, right? And one of those is going to work. And so like we're now in a place, right, where there's enough shots on goal that we've got a critical mass that one thing could keep moving us forwards, and then we create diversity around that, and we repeat. Um, so like I'm very grateful. I'm out of that hit or miss zone, right, where you're very much just fodder to probability, right? Like you'll be very lucky to make it out of that situation where you're like very narrow as a company. You've got one hypothesis, like that sort of stuff. So like we're in a much better place now.
That's a great life message. It's a great place to end it. I'm sorry to dive too much into technical stuff when there's so many good messages like that too. It's the professor in me, I think, wanted to pick your brain a little bit about that. But this has been a wonderful, wonderful conversation. Thanks so much, Daniel, for the great work you're doing and for spending time with us. And we want to have you back on the podcast soon.
You're very welcome, Robert, and thanks everyone for listening.
If you are enjoying this program, please hit that subscribe button, or even better, leave a review. Your support makes it possible for us to create the quality programming that we're continually striving for. Half of all heart attacks strike without warning. Your best early warning? A CT calcium scan. We've made it even better by adding arterial age and liver fat quantification, all from the same scan. Now available nationwide without a doctor's prescription. Use the code LUFKIN20 for 20% off. This episode is brought to you by L Nutra, maker of the Prolong fasting mimicking diet. Use the link in the show notes for 20% off. One of the most common questions I get asked are which blood tests I check for myself on a regular basis. Well, I check up to 50 biomarkers, but the secret is that I'm able to do everything from the convenience of my home, and I collect a sample with this new breakthrough device. So no more trays full of tubes of blood, needles, or even finger sticks, and you don't need to be a doctor to do it. Then I just mail it in for the results. SciFox Health makes it possible. Use the link below to get 20% off.
Can I start? Is it recording?
It's already recording.
Oh, sorry. Okay. This is for general information and educational purposes only, and it's not intended to constitute or substitute for medical advice or counseling, the practice of medicine, or the provision of healthcare, diagnosis, or treatment, or the creation of a physician-patient or clinical relationship. The use of this information is at their own user's risk. If you find this to be on the value, please hit that like button to subscribe to support the work that we do on this channel. And we take your suggestions and advice very seriously, so please let us know what you'd like to see on this channel. Thanks for watching, and we hope to see you next time. You should say that.
No, that's good. It's for you to say it. I think that's very good. You like it? You want to do it one more time, or is that good? I think that was good.
Yes.
All right.
You need to save the recording.